Dear colleagues, ladies and gentlemen, good evening. I'm Lina Pascu, Assistant Professor of Endocrinology from Athens, Greece. It is my great pleasure to welcome you in another ESE Spotlight of Science. Today, we're going to talk about new models of thyroid cancer. We have with us two excellent scientists, Inigo Landa and Hannah Nieto. I would like also to say that we are really glad to acknowledge that this webinar is sponsored by Recordat. Please allow me to say a few words about our speakers. I will start with Inigo Landa. Inigo Landa is a thyroid cancer scientist. Dr. Landa is currently a group leader at the Institute Gustave Roussy in Paris, France. He's also affiliated with INSERM U981 and the IHU National Precision Medicine Center in Oncology. Before that, he held an associate scientist position at the Division of Endocrinology at Brigham and Women's Hospital, and he was an assistant professor of medicine at Harvard Medical School in Boston, United States. He completed his postdoctoral training in Dr. James Fagin Lab at Memorial Sloan Kettering Cancer Center in New York, and his PhD studies in Dr. Robledo's group at the Spanish National Cancer Research Center and the Universidad Autónoma de Madrid in Spain. Prior to this, he got a double BSc in biology and biochemistry from the University of Navarra in Spain. The main focus of Dr. Landa's group is to identify key genetic events and to understand the underlying mechanisms in thyroid cancer progression. He is particularly interested in genomic alterations affecting gene expression. To this end, his lab employs a variety of techniques in preclinical cancer cell systems, genetically engineered mice and patient samples. So I think that we are really lucky to have Inigo with us today. He's the most appropriate person to talk about mouse models and thyroid cancer. And even if ladies go first, I introduced Inigo first because he will start first, but now it's time to say a few good words about Hannah Nieto. Hannah is a clinician scientist on a Birmingham Health Partners Fellowship at the University of Birmingham. Clinically, Hannah works as a consultant, ENT, hand, neck and thyroid surgeon at University Hospitals Birmingham, and recently completed a clinical advanced training interface group fellowship at the Manchester Royal Infirmary. Academically, Hannah is working in the field of thyroid molecular genomics with an interest in thyroid cancer and bioinformatics. Hannah delivers modules on statistics and bioinformatics at the University of Birmingham for interscalated students and supervises MSc anatomy students, coaching them through performing systematic reviews and meta-analysis. Her PhD was completed in the molecular biology of thyroid cancer recurrence, encompassing programming and wet lab discovery science. Hannah sits on the National ENT and IHR CRM. Her research focus is genetics in diagnostics and prognostics in thyroid cancer. It's amazing how a surgeon can combine research, basic research, and statistic research also, including meta-analysis. Hannah, really impressive. So, Inigo, Hannah, we really look forward to listen to your talk, and I'm sure that we will have questions at the end to discuss with you. Thank you.
All right. Hello, everyone. Thank you for the kind introduction and for the invitation to present here today. So we have prepared a combined presentation on new mouse models of thyroid cancer, both Dr. Nieto and myself, and we'll go back and forth at several points during the presentation. So we don't have any disclosures. So this is the outline of the presentation today. I'll give a brief introduction on the topic, and then we are going to discuss three papers, three mouse models, which I think are relevant on different aspects of thyroid cancer. The first one related to radioactive iodine treatment and redifferentiation trials. The second one is a good example on how to use models to discover mechanisms in metastatic disease. And the third one relying more on anaplastic thyroid cancer, immune infiltration, and responses to systemic therapies. So I don't think this audience needs an intro, but just a brief primer on thyroid cancer. Most differentiated thyroid cancers, or DTCs, are started by a single genetic event that constitutively activates the MAP kinase pathway. This is typically a mutation in BRAF V600E that leads to these DTCs, usually a papillary thyroid cancer. Those DTCs can progress into less differentiated and much more aggressive disease by acquiring a number of other events. And we distinguish, the metastatic and refractory DTCs that have lower survival rates. And at the other end of the spectrum, we have the anaplastic thyroid cancers, or ATCs, which are extremely aggressive tumors, heavily immunosuppressed. And this just to convey the message that this is a quite linear process, and we know most of the events leading towards this aggressive disease. Now, a couple of words on the genetically engineered mouse models that we'll be seeing here today. We're going to be seeing examples on two different types of models. Models that we call knock-in or knockout. In these models, we typically use a system that is the Cre recombinase, which is under the control of a thyroid-specific promoter, and that gives thyroid specificity. So this thyroid promoter in mouse models historically has been either thyroglobulin or TPO, thyroid peroxidase. So that drives the expression of Cre recombinase, and Cre recombinase has the ability to recombine this construct that is typically a tandem construct flanked by something that we call LOXP sites, in which we have the wild-type version of a gene and the mutant version of a gene. So when Cre is present in thyroid tissue specifically, that recombines, and we express the mutation of interest in the tissue of interest, thyroid in this case. There is a variation of this model, which are the inducible models, and it's based on several systems. Here I'm illustrating the ones that could be induced by tamoxifen. So the system is pretty similar, but here we have the ability to modulate the expression of this Cre recombinase at will. So we can switch on or switch off the Cre and then the oncogene or the mutation we are studying. So a general model or general approach for this kind of studies that we are going to be presenting. Typically, a mutation is identified by doing genomic profiling of tumors. Then we molecular biologists spend a lot of time in the lab engineering a mouse model, and those serve to assess the oncogenicity of certain mutations, to discover new mechanisms, or to test therapeutic combinations. And then the idea is being translational and eventually bringing that knowledge back to the patients. Now, let's start with the first part of the presentation, in which we are going to present a couple of papers related to how to use mouse models of thyroid cancer to understand responses to radioactive iodine treatment. So, as you know, the physiological function of a thyroid follicular cell is to concentrate iodine to make thyroid hormones, and that's determined by the proper expression and localization of the sodium iodine importer, or NIS. And this has been leveraged for decades for thyroid cancer treatment using therapeutic doses of radioactive iodine. And we've been using this for, I think, 80 years now, and this is like an old-fashioned paper from the 1940s, I believe. Now, jumping onto the first mouse model, This is a little bit of an older one, but I think it's important to understand the more recent research. Here's a mouse model of the inducible kind. So here, the authors were able to build this construct in which, by treating these animals with doxycycline, they were able to activate a mutant version of BRAF, and that's what they are seeing here. So when doxycycline was present, the oncogene was on, and the expression of V600E was present here. And then they had the ability to switch it off, and the oncogene was no longer expressed in the thyroid glands of these mice. The expression of BRAF V600E, as expected, led to constitutive activation of the MAP kinase pathway. And this is what you are seeing here in this Western blot, phosphomec, phospho-ERK, fully activated when the oncoprotein is on, and deactivated when they removed doxycycline and the oncoprotein was off. Now, in terms of thyroid gland biology, the activation of BRAF V600E, as expected now, we have this knowledge now, right, led to a massive enlargement of the thyroid gland. You can see after 1 week on of this oncoprotein activation, and then that went back to semi-normal size when they deactivated the oncoprotein. At the histological level, the overexpression of BRAF V600E led to the development of papillary thyroid cancers, some of which eventually progressed to poorly differentiated thyroid tumors. Now, another consequence, which I think is very interesting for the topic here today, of this activation of the MAP kinase pathway was not only cell proliferation and cancer induction, but also suppression of the normal thyroid function, including the expression of the sodium iodine importer. And this is clearly seen here. 1 week on the oncoprotein, you see a massive reduction of all of these thyroid differentiated function markers, including NIS. When you switch it off, NIS expression goes back on and stays on for a number of weeks. Obviously, that correlated with the ability of these animals to get some uptake of radioactive iodine in their thyroid. When the oncoprotein is on, the uptake is off. And when the oncoprotein is off, the uptake is partially restored. Now, if you can genetically switch on and off the oncoprotein, you can also pharmacologically play with it. And that's what they did using one of the first BRAF inhibitors, this PLX back in the day. I think now this is called vemurafenib. And they were able to restore the uptake of radioiodine in these mouse thyroids when they inhibited BRAF oncoprotein. So to summarize this data at the molecular point of view, so this part of the slide on the right, you have the normal TSH-mediated signaling in a thyroid follicular cell that leads to cyclic AMP, the recruitment of specific thyroid factors, FOXY1, PAX8, etc., that leads to the expression of these thyroid genes, including NIS, that is then expressed in the membrane and transport iodine. However, when we have constitutive activation of MAP kinase here on the left, in this example, by a mutation in BRAF V600E, there is an inhibition of this thyroid differentiated pathway at different levels. So NIS is not properly expressed. The obvious conclusion is we can block MAP kinase that eventually would lead to re-expression of NIS and proper uptake of iodine that we can use for radioiodine treatment here. And that's what is called redifferentiation, and that inspired or informed the first redifferentiation trial. And at this point, I'm going to switch to my colleague, Dr. Nieto, for the next few slides.
So yeah, I'm going to speak a little bit about the redifferentiation trials and give a little bit of a clinical kind of perspective on this. So as Inigo's outlined, you've got lots of different types of thyroid cancer, and differentiated thyroid cancer has really good outcomes, as we all know, but that's not just because of the disease, it's because we've got really good treatments for it. So we use surgery, plus or minus radioactive iodine when necessary. There are certain types of thyroid cancer which are much more challenging to treat, all kind of umbrellaed under advanced thyroid cancer, and there are lots of different definitions of what can be called advanced thyroid cancer. But essentially, it all kind of pans down to these nice diagram from Cancer Research UK, which is local invasion, so it's very difficult to resect, or has a lot of morbidity attached to it when you do resect it. Lymph node metastasis, which is just more surgery, even though it is generally resectable, or distant metastases, which are not necessarily resectable all the time. And then we become much more dependent on radioactive iodine. And then obviously there's anaplastic thyroid cancer as well, which is generally resistant to all kind of therapies that we have. But the kind of important thing to say with lymph node metastasis, especially if it's recurrence or metastases that aren't amenable to surgical treatment, is we become very dependent on radioactive iodine. And so when you have these young patients, often female, but around working age, they have 10 to 20% of them will go on to develop some form of radioactive iodine refractoriness. And when they become truly radioactive iodine refractory, if they've metastasized already, then they become an extremely challenging group to treat. And so I'm kind of just emphasizing here really that radioactive iodine resistance is kind of very important. So I'll just move on to the next slide, if that's okay, Inigo. And the idea then behind the redifferentiation trials is that we're going to take that information from the mouse studies that we've got, use medication, in this case MAP kinase inhibitors, to try and make the patients more sensitive to radioactive iodine therapy. If you can do another click. And the key really, as Inigo's already outlined, is when you redifferentiate, you get all those thyroid-related proteins that come back and are expressed more highly. And the most key one out of those is the NIS, sodium iodide importer, and because it allows us to use radioactive iodine effectively. The reason that this is so important for thyroid cancer is because it's specific to thyroid cells. And what that means is that radioactive iodine or destructive dose is only going to thyroid cells, unlike other chemotherapies or radiotherapies that we use for other cancers, which will affect all cells. So we're reducing the toxicity by getting a really, really specific treatment focused on the thyroid cells that we need to. If we skip on again. So what was really, really nice about the first mouse model study that Inigo has already described was that was the first kind of demonstration of MAP kinase inhibition that was leading to the redifferentiation, and it led almost directly onto this first selumetinib trial here that was published in the New England Journal of Medicine. Now, it was a kind of beautiful segue from kind of that mouse model into a clinical trial. They had 24 patients, they managed to follow up 20 of them. And what they did was they gave them recombinant TSH before an iodine 124 PET scan, which is what we normally do if we're scanning somebody. And then they had that scan both before selumetinib and after selumetinib, which was given twice a day for 4 weeks. Out of the 20, 12 people responded. And if they received a certain amount of a response on that radioactive iodine scan, they then went on, which those 12 patients did, to have therapeutic radioactive iodine. And out of the ones that had therapeutic radioactive iodine, 8 of those had a significant amount of volume reduction in their disease, and all of them had a reduction in their thyroglobulin, which is what we use to kind of track patients' response to treatment after they've had a total thyroidectomy and radioactive iodine. Nobody had any adverse effects apart from one patient who had myelodysplastic syndrome, which is about a year later. So obviously we can't say whether that was causation or not. And I'm going to go to the next slide, if that's okay. And that is to talk about the next trial that I want to talk about, which is all about RAF inhibition. I had another year where they were targeting a different type of the MAP kinase pathway with a RAF inhibitor. You can see they had a small number of patients that they followed up, 8 of whom had uptake on the radioactive iodine scan that they did. They did a 1, 2, 3 scan, I believe. And then 2 of those patients had a really good response to the radioactive iodine that they were given therapy therapeutically, and 4 of them had stable disease, have any response or continued to progress. So again, a kind of great step forward in terms of potential treatment for these patients that were getting no response at all. But we have got those patients who are resistant to this therapy for reasons that we haven't yet explored. Next slide, please. So what I've got here is a figure from the ITOG statement, which they've got quite a few nice tables in that study, which look at the different redifferentiation trials that they have with MAP kinase inhibitors. So it's not just these 2, there have been others since, and sadly selumetinib didn't look like it was going to have that much potential clinically on a wider basis, but it has led to many more trials with MAP kinase inhibitors that have had potential. And what we're looking at here is they've divided it up by background mutations. So you can see at the top, these are the BRAF mutations, and these are the bottom of the RAS mutations. And this is kind of reflective of that nice Cell paper from the TCGA a decade ago now, which started to split tumours up as to BRAF-like and RAS-like based on their tumour behaviour. And some of these also gave a little bit more of a breakdown about the mutational profile of these patients. And you can see I've highlighted one here study where 4 out of the responsive patients, 4 out of 6 BRAF patients had a response, but none with an ARID2 and none with an RBM10 mutation had a response. Now, those mutations are important, and we'll come back to them later on. And people are using mouse models now to unpick those genetic mutations and why exactly they might be conferring a resistance to the redifferentiation therapy in a little bit more detail. Next slide, please. So, redifferentiation insight so far. We've got a robust system of treatments for differentiated thyroid cancer, as we all understand. There's a huge challenge of radioactive iodine resistant disease clinically, and has potential for that to be addressed by redifferentiation therapy. There's a really, really nice transition from that mouse model to the first kind of clinical trials, and we're already seeing that we're getting a better response in RAS-like tumours, and we're going to look at the genetic background in a little bit more detail. There are clearly other means of escape from redifferentiation. Not everybody is redifferentiating the same way, and we're going to explore that next through the next mouse model. And I'll hand back over to Inigo, if that's okay.
Yeah, thank you, Hannah. So yeah, for the next mouse model, so the entryway into this data, and this was published back in 2021. So here I'm introducing you to this YSNF complex. This is a chromatin remodeling complex that is made up from multiple proteins encoded by several genes. And the key point here is that these genes are mutated in a number of different cancers listed here. Most of these mutations are loss of function mutations. So the proteins are no longer properly expressed. Oops, sorry. And that's the case in thyroid cancers. They are relatively infrequent in papillary thyroid cancers, especially in the most indolent ones, and they become more and more frequent when we profile poorly differentiated thyroid cancers and obviously anaplastics as well. So it's one of those events that is clearly enriched in advanced disease. Now, going to the paper, I'm showing you just some snippets of data. There's a massive amount of data on these papers. I'm just highlighting a few facts. So here what they used was a TP03 BRAF mouse model. They had the extra of being able to isolate the tumor cells because they were expressing this yellow fluorescence protein, or YFP, also under the control of TP03. And we know that these BRAF models, the induction of BRAF, leads to papillary thyroid cancers in mouse models, and that's what they report down here. Now, when they combined, when they combined BRAF with either the loss of ARID1A or ARID2 or SMARCB1, and those are 3 different components of this YSNF complex, they observed that these tumors progress to more aggressive tumors all the way to anaplastic thyroid cancers. Now, because YSNF is a complex involved in chromatin accessibility, that means which regions of the genome are preferentially open, and they have for transcription factors to have access to those and effectively activate transcription of the genes contained in those regions, they were profiling these tumors after they had isolated the thyroid cells from the microenvironment using a technique that is called ATAC-seq, which is basically a way of looking at the landscape of the genome and which regions are open and which ones are closed. And without going into detail, basically the disruption of the SWI/SNF complex by either of these truncating mutations led to a massive reconfiguration of the genome and closing of certain areas of the genome. Now, I want to highlight just one experiment from this mouse model. And what they did here was some kind of, again, a redifferentiation assay on these mice. They treated these mice, in this case with CKI, which is a dual inhibitor of MEK and RAF. And then they collected the tumor, they isolated cells to do their genomic approaches and a few other experiments. Now, if you focus on this lower part of the slide, and I think here the key comparison are the blue bars. The blue bars are the BRAF models, and the red, green, and purple are the BRAF. This is moving, okay. And the other colors represent the BRAF plus the disruption of either of these three genes from the SWI/SNF complex. And here we are looking at the expression of certain genes. This SLC5A5 is the gene that encodes NES, the sodium iodine importer, and then we have thyroglobulin, TPO, TSH receptor, etc. And you can see in this redifferentiation assay that after treatment with MAP kinase inhibitor in the BRAF model, those papillary with no other additional alterations, you can restore the expression of NES and a few other genes. But when you combine that with these extra mutations, not so much. And that's particularly striking for NES. That was confirmed by staining NES in these mouse tumors. And you can see how the staining was restored in BRAF, but not in the other models. And that correlated pretty well with the ability of these mouse thyroids to restore iodine uptake after treatment with MAP kinase inhibitors. So to summarize the data, we basically have a model in which, in a context of a metastatic thyroid cancer, BRAF-driven, treatment with RAF and or MEK inhibitors is able to restore the expression of NES in a condition in which this SWI/SNF complex is intact, is wild type. And that would eventually predict for the restoration of radioiodine avidity. However, when we have the same case, but on top of a BRAF mutation, we have a mutation that disrupts this chromatin remodeling complex, the chromatin region around NES is closed. So the restoration of this is not enough for transcription factors to kick in and be able to re-express NES. And with that, I'll hand it over to Hannah again.
Thanks. So we've got that information that certain mutations confer a resistance, particularly to redifferentiation therapy, on the kind of background of that we know that already BRAF-like tumours are much less responsive overall than RAS-like tumours. And this is a really nice figure from one of Inigo's papers that shows that possibly the reason behind that is that the RAS tumours are starting from an area of higher differentiation to start with, and so the recovery from that is slightly better in those particular patients. But clearly, I think not just the kind of RAS and BRAF differentiation, more genetic information is going to be absolutely key in terms of getting us a better understanding of who's going to be responsive to redifferentiation therapy. So if we can go to the next slide, please. So other people have been recognising this and exploring this. And so what this study that we've got up here is a retrospective case control study where people looked at some excellent responders to radioactive iodine versus non-responders and genetically profiled both and compared what the expression was between them. Now, unsurprisingly, we skip on a few clicks, Inigo, if that's okay. It will highlight the right areas. we can see that BRAF was enriched in a lot of the non-responders, and we can see that the other pathways, so the more RAS-like pathways with RAS mutations or translocations, are much more likely to be the excellent responders. And that was a really nice kind of paper to confirm that information. So next slide, please. So in terms of genomic characterization and insight so far, as Inigo said, we haven't by any means explored all of this so far, but we know that redifferentiation is more successful in RAS-like than BRAF tumours. There are other very specific mechanisms of resistance to redifferentiation. We know that in these patients, we sequence for targetable mutations, say in anaplastic tumours rather than radioactive iodine resistant tumours. But what we need to be doing is prognostication through sequencing, and it will come. And a lot of the commercial companies are trying to kind of get on top of this at the moment. What we want to do is be able to stratify patients with radioactive iodine resistant disease with extended characterization, not just those baseline mutations, and categorize how likely they are to respond to redifferentiation and even certain specific types of redifferentiation therapy. So Inigo is going to introduce our next mouse model, which demonstrates that genomic characterization is going to get us to understand these specific tumor behaviors in even more detail.
Very smooth transition there. So, yeah, I'm gonna briefly describe this paper that came out earlier this year, and it was specifically assessing the role of this RBM10 gene in thyroid cancer progression and in metastatic disease in particular. So RBM10, again, the motivation on why you spend 5 years making a mouse model, it always comes from the genomic profiling of patients. And RBM10 had been previously reported, specifically mutations, again, loss of function, and those are the black squares here, and the frame shifts here along the protein. In a genomic study assessing lethal non-anaplastic thyroid cancers. And there was some prognostic value there on assessing RBM10 mutant versus wild type in this cohort. This is a profile of the different the frequency of the different alterations on papillary thyroid cancers, the now newly defined differentiated high-grade thyroid cancers, poorly differentiated and anaplastics here in red. And RBM10 was one of the exceptions here. It wasn't always like a stepwise increase from PTC to ATC. I was particularly enriched in this intermediate disease, this metastatic DTCs, slightly more aggressive DTCs, not your usual papillary thyroid cancer confined into the thyroid gland. So RBM10 is a protein. RBM10 stands for RNA binding motif protein 10. It's encoded in chromosome X, and it had previously been reported to have a function on alternative splicing. Now, splicing, as a reminder, is the process by which we can edit precursors of messenger RNAs into multiple isoforms. And this is a figure for a nice review, showing the many mechanisms of alternative splicing, from the use of alternative promoters to inclusion and exclusion of certain exons. And this is a mechanism that cells use to expand the proteome. And they tend to be very tissue specific and also cancer specific. So going to the paper, again, starting with the model. Here, what they used was a model of in which they were able to induce, in this case, the HRAS G12V oncoprotein using this TPO-CRI system again. And on top of that, they were able to abrogate the expression of RBM10 using a fluxed allele also under the control of TPO-CRI. And that's simply how they show that the expression of RBM10 is no longer present in those animals with both HRAS and RBM10 mutation. HRAS is notoriously weak oncoprotein, particularly in mouse models. and actually it's hard to generate a tumor by just inducing HRAS. You need to wait for at least a year, and mostly you get hyperplasia, and that's what they observed. And now when they combined HRAS with loss of RBM10, they started seeing this acceleration of onset of papillary thyroid cancers that could progress to anaplastic thyroid cancers with a longer latency, and eventually these animals generated lung metastasis. So in other words, you are generating a nice model of metastatic PTC, primarily driven by the loss of this RBM10 protein. So you are proving experimentally that RBM10 loss associates or can promote metastatic behavior. Let's see if it moves forward. There you go. Now, again, I'll just give a brief summary of the paper. They went ahead and on top of this mouse model, they use human-derived anaplastic thyroid cancer cell lines to run a number of experiments, and they had this nice approach in which they would use models with endogenous mutations in RBM10, and in those the expression of the protein was no longer present, but they could silence the protein, and the other way around. Sorry, I'm doing this in reverse. The endogenous mutations are down here, and here they have the overexpression models. So with those cell lines, they generated, a number of what we call like paired cell lines or isogenic models, and they were able to profile the specific RNAs that are being produced by those models, and they run a number of other experiments. So this is the summary of the profiling that they did in those cell lines with or without proper RBM10 function. And when those are the different transcripts that were overexpressed and underexpressed. And when they did a pathway analysis of those specifically significantly overexpressed and underexpressed transcripts, they saw that most of the processes had to do with cell motility, cell migration, extracellular matrix. In other words, the ability of these cells to proliferate, to move around, to invade, which makes sense because as we observe on the mouse models, this is conferring a metastatic ability to these thyroid cancer cells. So they focused on three targets, which are highlighted here. Vinculin, which is a modulator of focal adhesion, CD44, which is another cell surface adhesion marker, and it's involved in cell-cell and cell matrix interactions, and tenascin C, which is an extracellular matrix protein, and it binds fibronectin. Now, it is important to highlight here that they were looking at alternative spliced targets. So these versions of these proteins have certain exon inclusions or exclusions. So they are kind of like unique versions of these messenger RNAs and proteins that are preferentially being transcribed and produced and translated in this cell context in which RBM10 function is not wild type. So they ran a number of experiments related to migration and invasion capacities of these cells, first in vitro, and they were able to demonstrate that switching on and off those specific aberrantly spliced versions of those three targets that they focused on could revert or enhance the phenotypes that they were seeing. And they also generated a cell line from the mouse model that they generated, the HRAS RBM10, and then they injected those in syngenic models of the disease, and they observed that when RBM10 was not present, they would generate lung metastasis, and that's shown down here. And then they could restore the expression of RBM10, and they would revert the phenotype, or they would silence this alternatively spliced transcript, and they would also revert or rescue the phenotype. So it's kind of like proving that what we see in terms of biological behavior is directly related to the genetic status of these animals and cells derived from these tumors. They went ahead and they also observed that all of these three targets converge in the activation of RAC1, which is an important protein in cell signaling, and that's why they focused on this, and they were again able to see activation of that protein that could be reversed in all of these human and mouse cell models, depending on the status of RBM10 being intact or disrupted. Switching back to you, Hannah.
So we're talking here about metastatic differentiated thyroid cancer, and I think as Inigo has kind of highlighted, this is different to just a dedifferentiated thyroid cancer. So there are different genes at play here. So we often think now, since Inigo's paper in 2016, we know that we have differentiated thyroid cancer and the acquisition of mutations all the way down to anaplastic thyroid cancer. And but for these particular tumours, they're not necessarily hugely dedifferentiated, or they might be a little bit, but they are metastatic. And so this figure was just to put up the fact that there are other factors that contribute to metastasis that are not just purely dedifferentiation, which is why it's really interesting when these particular genes come up, especially something like RB10 that has such an impact on other proteins in the cell because it's affecting splicing. Next slide, please. So as we've already discussed, there was a distinct kind of profile of different extracellular matrix proteins, so things that relate to the tumour microenvironment that were modulated in that paper, including vinculin, CD44 and fibronectin, which are all classic ECM proteins. So that's interesting from that perspective, and it's going to be interesting to see if any of these can be used in prognostication, because they're a very well versed kind of immunohistochemistry stains for most of these proteins. Next slide, please. So one of those proteins that we were talking about from that mouse model was tenascin C, which was, highlighted by the RBM10 mouse model, and that's been taken forward by a different group here. So Hartman et al. Have been looking at this in a little bit more detail. And it's a really interesting protein. It binds with fibronectin, which I think Inigo mentioned, and you can see that it's associated with lots of different aggressive thyroid behaviour. So there's a nice chart here showing that it's overexpressed in lymph node metastasis compared to the primary tumour, and the IF slide here shows that the tenascin C is shown at the kind of leading edge of the tumour. So they've done lots of studies staining for this, and they can see whether tumour cells are making progress forward. That's where it's most highly expressed. So associated with lots of aggressive tumour behaviours, but specifically with tumour migration and all the things that lead to tumour metastasis, which just kind of demonstrates another kind of strength of the mouse models, that they are allowing us to investigate genomic characterization of these particular mutations that relate to metastasis rather than just general dedifferentiation in thyroid cancers. I'll go on to the next slide, if that's okay, Inigo, which I think is back to you.
Okay, back to me. Right,
Yeah.
so, oh yeah, sorry. Yeah, you froze for a moment. So let's go with the third example of the presentation. So this paper was published a month ago. There's not even like a finalized PDF out there. So it's still the manuscript version. So if you wanted a novel mouse model, this is the most novel you can get as of September 2025. And I would highly recommend that you go through this paper. Not an easy read, but I think it's a tour de force in this specific field. Done by full disclosure by my former boss, postdoctoral mentor. I was not involved in this paper whatsoever. So now, here we're focusing mostly on anaplastic thyroid cancers. And what I'm showing you here from this paper is basically a characterization of the tumor microenvironment, and specifically the immune infiltrate of these tumors. As you know, they are infiltrated heavily by immunosuppressive cells. So the part on the top is human data. This is from a tissue microarray on papillary, including papillary, poorly differentiated and anaplastic thyroid cancers. And what you are seeing here is a characterization by multiplex immunofluorescence and looking at the different immune cell subpopulations. And this is the quantification of that data on the left. So the main conclusion here is that human anaplastic thyroid cancers are enriched by macrophages as expected, and specifically by M2, the immunosuppressive tumor promoting macrophages, as well as by T cells compared to papillary thyroid cancers. Now, the data on the bottom is the equivalent characterization of the immune infiltrate of tumors, but in this case, showing data from mouse models. And here they are comparing two models, a papillary thyroid cancer model, which is basically a BRAF-driven tumor. Again, thyroid specific. I think this is a TPO-CRE model again, a different version of that model. And on the right, you have an anaplastic thyroid cancer model in which on top of BRAF activation, we have loss of function of p53. That, as you probably know, is one of the main triggers towards PTC to ATC progression. And the conclusion here is that these models are representative of the human tumors. You also see a high degree of infiltration in anaplastic thyroid cancers from mice of T cells, specifically the CD4 population, as well as macrophages and other myelosuppressive cells quantified here. Now, a few highlights from this long and very interesting paper. So here they were using two different mouse models of anaplastic thyroid cancer. On the top, this is a doxycycline inducible model. We briefly went over this model at the beginning, but on top of the BRAF oncoprotein, they also had loss of function of p53. So the key here is that by giving these animals chow with or without doxycycline, you could, as we saw at the beginning, switch on or off the oncoprotein, the MAP kinase pathway, right? So in a way, you are doing genetically what you could also do pharmacologically with a MAP kinase inhibition. And this is important in the context of anaplastic thyroid cancers, especially those, well, actually those that are BRAF V600E driven, now being typically treated with dabrafenib and trametinib, RAF inhibitor and a MEK inhibitor. So here they were doing that genetically, inactivating the protein. The model below is a different model of BRAF activation and p53 inactivation. And from that model, they would generate a cell line, this TBP3743, and then they would orthotopically inject those cells in the thyroids of mice of the same background to make sure that the immune system was fully competent in those animals. And with those, this is not properly displayed here, those animals with those anaplastic thyroid cancers in their thyroid glands, orthotopically, they would be treated with either vehicle or dabrafenib and trametinib, so inhibiting MAP kinase pathway. And then they isolated the thyroid cells, and then they did RNA sequencing on these two equivalent models in which you switch off and off MAP kinase. And what they observed in both models, and those are highlighted here by these color boxes, is that MAP kinase inhibition led to an enrichment of antigen presentation and interferon pathway activation in thyroid cells within those anaplastic thyroid cancer models and these specific BRAF and P53 loss contexts. Now, looking at a specific or digging deeper into the determinants of that antigen presentation pathway, they observed that the inhibition of MAP kinase induced expression of genes involved with the major histocompatibility complex 2 in those anaplastic thyroid cancer cells. And here they highlight a number of genes that are encoding in the mouse genome that MHC2 complex. And they focus their research on this CIITA gene, which is one of the master transcriptional regulators of major histocompatibility complex 2 related genes. And they did an extensive amount of experiments on this specific factor because that triggers the expression of all of this pathway. Here on the bottom, this I think requires a little bit of an extra explanation. They were using 4 different cell lines that they derived from these anaplastic thyroid cancer tumors, from this work and from previous work from the same lab. And here they divided the cell lines into groups. The ones in orange are the ones that are primary, that after MAP kinase inhibition, they retract. The ones in green are relapses. And this was coming from a previous study in which some of those relapses could be attributed to other factors, such as overexpression of or amplification of MET and a few others. But the interesting point here is the different behavior of these cell lines when they were subjected to MAP kinase inhibition. In this case, they were treating this cell with interferon and trametinib, which is a MEK inhibitor, and they would saw that restoration or that overexpression of these markers of this MHC2 pathway in those primary cell lines, but that expression was abrogated in those that relapsed, suggesting that the expression of those factors are important for those responses or for the most dramatic responses to MAP kinase inhibition in this specific BRAF and P53 anaplastic thyroid cancer context. They went further, and I'm going to skip this for the most part, but they were able to demonstrate, again, via RNA-seq and ATAC-seq, that this master regulation, the CIITA, was epigenetically silenced in those genes, in those cell lines in which they were recurrent, that were coming from relapses. So here what they are showing is this loss of peaks of accessibility for STAT1, which is a regulator of this master regulator of MHC2 component. And you see how those peaks are lost in exclusively after treatment in those recurrent cell lines. To wrap up, or at least the highlights of this paper, they went ahead and they tried to demonstrate how or whether these MAP kinase inhibition responses are primarily mediated by this MHC2 and CD4 subpopulation of T cells. So here on the top, they were showing in these orthotopic models of injection of ATC cells, how they would, and after treatment on dabrafenib and trametinib, how these tumors would become heavily infiltrated with both CD4 and CD8 positive T cells, and that's quantified here, vehicle versus MAP kinase inhibition. However, they wanted to further dissect the contribution of each of those CD4, CD8 subpopulation, in other words, MHC 1 and 2, 2 and 1 actually. And so what they did is with those cells, they not only implanted the cells that I just mentioned, but they also edited via CRISPR those cells to here abrogate the expression of this CIITA regulator, as well as a different gene from this MHC2 encoded mouse program. And they observed that the dramatic reductions on tumor volume here is the comparison of vehicle-treated versus dabrafenib and trametinib-treated mouse tumors. They were abrogated when these two master regulators were not present, directly linking the responses to dabrafenib and trametinib to the expression or the restoration of expression or the overexpression of those specific factors. They also observed that if you deplete these animals from CD8 positive T cells, the effects are maintained, but CD8-4 positive T cells are required for those responses to be present in anaplastic thyroid tumors driven by BRAF and P53 in the context of dabrafenib and trametinib inhibition. Back to you, Hannah.
Thanks. So it's a bit of a masterclass, that paper, isn't it, in mouse models and lots of other techniques. So systemic therapies, insights so far. We've got clinically well used like dabrafenib and trametinib therapies for anaplastic thyroid cancer, and we have emerging treatments or patients that can go into solid organ trials for specific mutations that they have. But no matter that, even when we're using combined therapies, we have like escape from this treatment. And that's often what's limiting these patients. At some point, they gain another mutation and then their disease progresses later on, even if they get a relative amount of time from this treatment. Next slide, please. So when Jim Fagan presented this at the ATA, the paper that Inigo's just been going through, what he was saying, his kind of overview or insight from that paper was that in the future we'll potentially be treating with MAPK inhibitors to make that really immunogenic environment really responsive to the immune checkpoint blockades drugs that we can use. So maybe using those in combination is the thing that's going to actually get these patients either into remission or into a point where they are surgically resectable with these cancers. Yeah, next slide, please. So our conclusions really are that thyroid cancer mouse models are incredibly useful. They prove the oncogenicity of genetic alterations, both in initiation of tumour and progression. They help us understand the thyroid cancer cell biology, interactions with the microenvironment. They're a great mechanism for testing therapeutic approaches in those kind of preclinical studies. There are limitations and challenges as kind of listed here. Where are those oncoproteins functioning? Oh, go back again, if that's okay. There's certain genetic mutations that are very difficult to model. And obviously there are intrinsic differences between mouse and human biology that we know. However, I think our kind of overview of this is proving how incredibly useful mouse models have been. And it's really nice to see those kind of, not only those mouse models that have kind of directly impacted clinical care in the past, but also these really, really up-to-date mouse models that are going to affect how we're treating patients in the future, potentially. So the next slide is just a barcode really linking you to a really nice paper by Jeff Nauf, who's done, and the other author Dutta there, who've done a really nice overview of the most recent mouse models in thyroid cancer. And there's lots of kind of table summaries in there, and it's subcategorized by the different genetic mutations they've looked at. So it's a really nice paper if people want to go away and read about more mouse models apart from the ones that we've outlined here already. And that was published this year, I think, as well. And that's the end. We'd be very happy to answer any questions that anybody has.
Thank you, Hannah and Inigo, for the very nice lecture. I think that we really enjoyed it, and you proved how a scientist, a basic scientist, can collaborate with a surgeon for a great result. And I think that thyroid cancer shows us how we can really work all together for the good of the patient at the end. So, Inigo, do you have any comments for conclusions or highlights?
I would like to, stress the fact that mechanistic studies are by definition a little bit behind the discovery of a specific determinants of aggressive disease, but are absolutely necessary. And they are the link between what we see in patients' tumors and a potential clinical application into that. And that requires, as you mentioned, lots of collaboration, lots of different skills put into the same question from a different perspective. And it's an exciting moment to work on this, not only on mouse models, but on organoids, on any other kind of more and more refined avatars to study specifically aggressive disease.
Thank you very much. And Hannah, how do you find to do basic science in between seeing your patients or collaborating with basic scientists? It's a more general question, but it's good for younger people to listen to these things.
Challenging. Yeah, I mean, there's never enough time for either side, and I certainly find that the clinical work tends to eat up time if you let it kind of get away with it. I'm fortunate. I did my PhD with the McCabe group in Birmingham, which are incredibly strong kind of discovery scientists, and Chris is very used to having clinicians through his lab and teaching us good science. And then what I'm kind of excited to do next is start to hopefully branch into some more clinical trials. Because I think if I'm doing that, then I can really hopefully embed the mechanistic parts that need doing alongside those, because you're obviously getting a lot of patient samples in that you can answer a lot of kind of discovery science questions with at the same time. So that's how I'm kind of hoping to move forward with the next steps. But yeah, it's easier when you're doing a PhD. You can step away from the clinical side and just do the science, but kind of merging them back together, it is tricky, but lots of opportunities and very exciting when you can see that discovery science coming through to the bedside. It's nice.
So PhD is something that you recommend to all medical doctors that really want to go into science. This is what I understand.
Yes, I think it's always great to take time out of clinical training to do some kind of research or do something else that you're interested in. I think if you want to become, I think doing a full PhD is probably something for clinical academics rather than for every surgeon.
Thank you. So if there are any questions, we can conclude. Dear colleagues, ladies and gentlemen, thank you again for attending this very interesting ESE Spotlight on Science. Hannah, Inigo, thank you very much for your lectures. I really hope that you all enjoyed that. And the next ESE Spotlight of Science will be in November, but until then we have some other interesting scientific events that take place. You can see that in this slide, and I think that European Society of Endocrinology is keeping us busy. There are many events to attend and learn for everyone. Pure clinicians, pure scientists, or people that want to combine. We're really glad again to acknowledge that Ricordati has sponsored this event, and thank you all very much. Have an excellent evening. See you soon.
Thank you.
Thank you. Bye.